Thank you. I'm going to talk today about KRAS-BRAF as a vital tool for cancer therapy. At AutoGenomics, we have an infinity system in which we can analyze gene targets in a multiplex fashion and identify many different mutations simultaneously. So our initial goals were to identify mutations at codons 12, 13, and 61 in KRAS, and also codon 600 in BRAF. We wanted to work in a single tube format. We wanted it to be performed on our Infinity Analyzer, and we wanted to make sure that we could use the relevant specimens for assessing mutation in different cancers. And this includes formalin-fixed, paraffin-embedded tissue biopsy material. So after we reviewed the relative frequency of mutations for codons 12, 13, 61, and also in BRAF, we came up with a test panel, which included being able to detect 7 mutations in codon 12, 6 mutations in codon 13, and another 6 mutations in codon 61. In addition, we identified 6 mutations in BRAF gene V600 that we also wanted to detect in this assay. So the specimen types that we were using in development of the assay and also post-development include DNA extracted from the formalin-fixed, paraffin-embedded biopsy material, fresh tissues, frozen tissues, material that's scraped off of histology slides, and then also a number of cell lines that we use for controls to verify that the assay is working as we would like it to. So basically the workflow that typically happens when you're running a KRAS-BRAF assay is that a tumor sample is taken, it's sent to a laboratory, and then it's fixed and embedded in a paraffin wax. A pathologist then slices sections off of that block and looks at the sample under a microscope and assesses whether or not there are cancer cells present in that slice of tissue. Once the cancer has been determined by histological means, then oftentimes the lab will then extract DNA and then use it in a KRAS-BRAF assay. In our case, it's a multiplex PCR reaction to amplify the targeted regions of KRAS and BRAF. And then we would run that on our Infinity Analyzer to determine which mutations were detected for both KRAS and BRAF. The assay itself is designed to work with only 20 to 60 nanograms of input DNA. It's a single tube format for amplification, and that goes directly on the Infinity. It detects over 20 mutations, and it also detects the wild type sequences. So oftentimes biopsies contain both normal and cancer cells, so it's important for us to be able to measure both simultaneously in this assay. And it works well with DNA extracted from the typical biopsy material, which is in formalin-fixed, paraffin-embedded blocks. At this point, I'm going to go into a little bit of data with the assay. So one of the nice things is that there's a number of cell lines which have already been characterized for whether or not they have a mutation in KRAS or BRAF, and those were available to us to help develop the assay and also serve as controls for us as we run the assay. Here's an example of the signal that's seen for a wild type cell line. You see in our assay, almost all of the signal for codons 12 and 13 are in the wild type channel, Q61 wild type channel, V600 wild type channel. Now, using a different cell line that has a mutation in codon 12, the glycine to valine mutation, what you can see is that this is a homozygous cell line. It has all of its signal really in the G12 valine channel. Q61 is wild type, V600 for BRAF is wild type. Looking at a heterozygous cell line for BRAF V600E, all of the wild type signals in the G12 and G13. Q61 is all wild type channel. And what you see is almost an equal molar amount of wild type V600 and mutant V600E in this sample. This tells us that the assay is capable of picking up mutations and the normal signal also within the samples. We've done mixing experiments, controlled experiments. We've taken DNA from a mutant cell line and from a normal cell line, mixed them together, And what we can tell is that we have an analytical sensitivity that comes down all the way to 1%. So we can pick up below 1% cancer cells in a mixed cell sample. This is an example of the readout that you see on the Infinity. We have 3 different amplicons, one for codon 12 and 13, one for Q61, and one for BRAF. And what the software will do is, based on the amount of signal that's detected, it will give you a positive or negative indicator for the presence or absence of a mutation. And then at the bottom, it's summarized mutation detected for G12A in this case. We've done a number of comparison studies, and in this one case, we tested over 60 samples, and we saw a very good concordance with sequencing and the picking up mutations in the Infinity assay. In addition, we've seen better sensitivity than straightforward sequencing, and we pick up more mutations than present in some of the other RT-PCR approaches. So to summarize, this assay is developed to be easy to use. It's an automated assay, detects over 20 mutations. It works well with FFPE tissue samples. It's 1 tube per sample, uses only 20 to 60 nanograms per reaction. Our analytical sensitivity is as low as 1%. And our preset readout and summary set values are for detecting 10% mutation signal per amplicon. Now, this assay is a research use only assay, and it runs well on the Infinity. The next thing I want to talk about is that this pathway, EGFR, the EGFR pathway, plays an important role in a variety of cancers. As such, there's a number of different targets within the pathway that research is being done now to ascertain whether or not they're effective drug targets. Now, if we look at this, we've looked at RAS and RAF. There's additional MEK gene, MAP kinase. There's an alternative pathway through PI3 kinase. This is a very complicated pathway. As such, it leads us to believe that there will be additional opportunities for detecting mutations in other genes. So 2 of the areas that we're looking at right now include a expanded panel for BRAF that picks up codons 597, 601, and 600 that are in the activation domain for BRAF, and then 3 other codons where mutations have been shown to be important, which are located in the P domain. So this is an expanded panel for BRAF. This is still in the research stage, but it picks up a number of different mutations in the BRAF gene alone. In addition to that, we've created another assay which is picking up 10 mutations in exon 18 of EGFR, exon 19, 18 mutations, exon 20, 17 mutations, exon 21, 6 mutations. So in this assay, which again is still in the research stage, it picks up over 50 different mutations simultaneously. So we're expanding our number of assays to include mutation detection assays for different oncogenes, and these are 2 examples. So at this point, I'll turn it back over to Rob, where he's going to talk about women's health.
Okay, well, I'm back again. We get to talk about another topic. This is a topic